Welcome back. So, we are continuing our discussion on the applications and emerging research areas in neurophotonics, and today I have got a very interesting topic, that is nanoparticles for optical modulation of neuronal behavior. So, this is the essence of nano neurophotonics or nano optogenetics, the true heart of nanobiophotonics. Just to recapitulate, what exactly is a nanoparticle? So, nanoparticles typically range from 1 to 100 nanometer in size. We have discussed this in a total module, but I thought that couple of slides will help to recapitulate. We are interested in nanoscale because high surface to volume ratio, the optical property of nanomaterial depend on their size. So, if they are of the same material, silver nanoparticle, but of different sizes, they can emit or reflect, depending on the type of nanomaterial, different types of light. So, silver by definition bulk is whitish in color, but as you go on changing their sizes in a nanoscale level 1 to 100 nanometer, they keep on showing their individual distinct color properties. Nanoparticles can be made from various materials, polymers, phospholipids, inorganics, and of course, metals. What are the benefits of nanomaterials? Well, obviously, they are nano. So, nanomaterials interact with biological system at the molecular level. They penetrate the blood-brain barrier to deliver agents to targeted cells. Remember, we discussed about blood-brain barrier. Blood do not go directly inside brain. There are no capillaries, no veins, no arteries inside the brain. There is a semi-permeable membrane between brain and blood. So, it is specifically there so that any infection in blood do not get directly to the brain. Brain is thus far, since the most important organ, is protected fully. So, that blood-brain barrier can be penetrated by your nanoparticles, right. This is a 30 nanometer gold nanoparticle transmission electron image. See how small these are. We are presently working with gold and silver nanoparticle, but our work is for detection of viruses and proteins, but they are fascinating these days. This is the current flavor. I am myself fascinated by nanoparticles. So, real time monitoring of biological processes at single cell level possible. Optically active nanoparticles enable precise cellular interventions. They are very small, so they can affect very small area, and very small area, even though affected, can trigger some very big biochemical modification. So, you can understand what is going on. So, what are the optical effects of nanoparticles? So, mechanism based division of nanoparticles. It is photothermal, light falling on nanoparticle generating heat. Photomechanical, light falling on nanoparticle generating some kind of a mechanical change, pressure, etc. Photoelectric, light falling on to nanoparticles generating electric current. And photochemical, light falling on nanoparticle is triggering a biochemical reaction. So, be absolutely aware of these 4 categories and what they actually mean. So, nanoparticles and photothermal properties. A variety of nanoparticles exploit photothermal effects. Gold nanoparticles are notable due to optical response, stability, low toxicity and versatility. They are low toxic. Usually gold do not react with anything else. So, it will, if you have inserted gold nanoparticles inside, this is gold, yeah, it is not yellowish, this is nanoparticle, so it can be reddish. So, if you have inserted inside a cell, it will probably not react with its environment chemically, right. And when you have shine light on it, the electrons inside can agitate and create some sort of temperature difference around it, few Kelvin temperature difference, but good enough to modify the few nanometer cube volume around which around the periphery of this individual nanoparticle. Surface functionalization with ligands. Ligands are cofactors are type of ligands. There is some sort of a similarity, but ligands are basically something that binds with it can be anything, it can be proteins as well. So, ligands are those materials that binds, enable specific interactions with cells and tissues. So, gold nanoparticles and photothermal mechanisms. Gold nanoparticles can create surface localized surface plasmon resonance. I told you that metals have this electron cloud, and these electron clouds are moving at a very high frequency, plasma frequency, and whenever there is a external electric field, the localized surface plasmons go there and try to screen it, and in essence generate vibration in molecules. LSPR generates coherent oscillation, where energy transferred as thermal energy to the medium. The peak of the wavelength, the peak of this localized surface plasmon resonance, these electrons which are at the surface activated because of the free electrons of the cloud of metals which go to screen external electromagnetic field at the surface, LSPR, localized surface plasmon resonance. The peak wavelength depends on particle morphology, distance, and surrounding medium's refractive index. So, you can control them. They are highly effective due to small size. So, basically you are generating light to create specific amount of heat in the nanoparticle, and that is affecting its surrounding areas. AUNP heat immediate environment, allowing reduced overall heat delivery, sub-millisecond time scale, crucial for neuronal simulation, accurate targeting, minimize off-target heating and toxicity. Another thing that is quite interesting and has recently caught scientists' eye is formation of vapor bubble. So, this is fascinating. Think about it. I will try to explain it. Both are more or less same, plasma induced bubble or thermal induced bubble, but this is something that we discussed in that photoacoustic microscopy. So, you have a nanoparticle, you are sending light into it, the electrons inside are agitating and whatnot, and they have created some sort of a heat inside it. That heat is heating, thermally heating the surrounding area of the nanoparticle. Usually, as I said, the surrounding area is water, surrounding area is water or waterish. So, they can simply heat up and become some kind of a steam, but this steam bubble is surrounding the nanoparticle. The nanoparticle has heated up, the nanoparticle in terms the heat has transferred to its surrounding area. The surrounding area is water, the water has heated up and become steam, water vapor, it has become steam. But the steam by itself is semi-insulating, meaning the steam is bound to the nanoparticle, and if the nanoparticle is traveling, the steam is traveling with it as long as the nanoparticle is heated, right. Heat usually is not like this, as soon as the light heat comes up and it goes down, it takes some time to dissipate. And this nanobubble, because of its semi-insulating nature, does not allow any other area to heat up, does not allow the heat to transfer, does not allow any other heat to go in. It is semi-insulating, water vapor is semi-insulating in nature, and it simply moves around, or it is a localized heating environment that goes inside the cell. So, if you are targeting a particular organelle to be destroyed, a particular protein to be destroyed, this is one of the best options. And this is fascinating. Rapid heating induced evaporation and plasma formation. Rapid heating induced evaporation led to vapor bubble formation without significant thermal heating of the environment. Plasma formation mechanism observed under longer laser pulses and involves thermal heating of cellular environment. Non-plasmonic nanomaterials like carbon black can also generate vapor bubble through laser activated chemical reaction. So, there is a vapor surrounding the nanoparticle, and it can move, as I said previous in the photoacousto-optic lecture, that it could be considered as a mechanical wave. Mechanical waves, rarefication and contraction could be considered as sound, but nevertheless here very small nanoparticles can be created to generate this vapor bubble, and then this vapor bubble can be utilized to target very small, very specific areas inside the cell, inside the neurons, inside the dendrites, inside the axons to affect specific areas, right. Applications, see, this is exactly what I was telling you. Vapor bubble causes pore formation in cell membrane termed optoporation, through expansion induced hydrodynamic stress or liquid jets. So, basically you are breaking this down, you are melting just this part without affecting any other area, right. You have melted this area. Optoporation, modulating drug intake by neuronal cells, line with 3D plasmonic nanostructure, cell transfections without viruses, concern including prolonged pore closure and potential cell damage. Yeah, this is something that takes place. Photoelectric effect of nanoparticles. So, this was photomechanical effect. This is the heat causes the mechanical steam wave to pass through. Photoelectric effect, photoconductive materials like silicon and conductive polymers generate electric field under optical simulation, you know this. Nanoparticles with photoelectric properties are being explored for spatially selective neuronal stimulation. So, action potential is generated by the presence of a nanoparticle, because when light is falling into the nanoparticle is generating electric current. That electric current can be considered as an electric action potential, triggering action potential, resulting in the neuron getting fired. There are plethora of nanoparticles. You have quantum dots, tunable optical properties. You do not necessarily have to use metals. You can have organic materials like graphene. These days graphene quantum dots are quite popular because it's made up of carbon, and carbon is supposedly more biocompatible, but everything depends on dose, how much of graphene you are putting. Reduced graphene oxide nanomeshes are used to modulate neuronal cell. Carbon nanotube generates photocurrent when stimulated by visible near infrared range. So, all of those semiconductor and metallic nanoparticles, all of those can be inserted inside. So, their advantage is A, they are small, B, they are, because of their small, have less cytotoxicity, cellular toxicity, B, they can produce a plethora photothermal, mechanical, photoelectric, etc. All those mechanism of action. Neuron and quantum dot interface. Quantum dots generate electric fields. So, they generate electric field, not necessarily electric current. Electric fields allows the attraction or repulsion of specific ions, positive ions or negative ions in specific direction, resulting in free electrons and holes upon optical simulations, resulting in either resting potential or action potential getting triggered. So, neuron quantum dot interface is something that is coming up very strongly. Quantum dot we have discussed, very small particles, 0D, 0 dimensions, and they can be used to target very specific locations, right. What are the nanoparticle driven photochemical processes? Nanoparticle with photoresponsive group induced chemical changes within their structure. Common changes include isomerization, dimerization, combined together, isomerization, the cis and trans, all those things are changing, and bond cleavage reaction. UV and blue light trigger rapid structural changes, whereas near infrared response can also induce photochemical responses through two photon events, and up conversation non-linear processes can take place. So, obviously huge challenges remain. This is much easier said than done. How do you precisely activate one nanoparticle here, not other nanoparticle in the periphery? how do you target a specific nanoparticle at specific cellular or neuronal areas, nonlinearity effect, whenever you have excited gold nanoparticles, say for example with the light, which one of these four, photothermal, photoconductive, photomechanical or photochemical, do you think is going to happen? It may so happen that more than one effect plays together simultaneously. What then? It's very uncommon that only one thing will happen, right? Several of these can combine together. You both generated heat as well as generated mechanical wave, as well as it targeted some kind of a electric current. At the same time, it has created some kind of a biochemical reaction. It has triggered a biochemical reaction. So, we have to create some of these nanoparticles which are very specific for specific purposes. Some caged compounds interact with receptors before photoactivation, requiring further development. So, 2D materials are coming up very strongly, but this is a completely unexplored area. So, those of you who are from chemistry or biochemistry, chemical engineering field, I would recommend you to take on these nanoparticles that modify neurons, nanoparticles that modify neurons. 1001 application against optogenetic enhancement, neurostimulation, neural circuit mapping and neural tissue engineering. Several of these are there, read it at your own leisure time, and these are the conclusion. The integration of nanotechnology and neuroscience presents a fascinating frontier that has the potential to reshape our understanding, and the more people such as you come and work at the interface of nanotechnology and neuroscience, nano-neuroscience, think how fascinating it would be, where not only you will be understanding the brain from a comprehensive standpoint, but you also will be able to manipulate it using nanotechnological materials, tools, instruments, etc. Right. So, these are the concepts covered and these are my references. Thank you very much.